Good afternoon. Welcome to Water Research Foundation webcast, Guidance Manual for Monitoring Biological Filtration of Drinking Water. My name is Kenan Özekin. I will be the moderator for today's presentation. The Guidance Manual for Monitoring Biofiltration, Monitoring Biological Filtration of Drinking Water Project 4620 was funded under the Biofiltration Research Area and focused on practical biofiltration monitoring tools that provide rapid feedback on the performance of biological filters. During the webcast, research team will present data from the study and provide information on various monitoring tools. Before we start the webcast, I have a couple of administrative items. There will be a brief question and answer period after the presentation. You can ask your question at any time during the presentation by typing your question on the web page. If you are unable to get everyone's questions today, we will provide written answers for those questions that are not answered. In addition, the webinar itself, including the slides, will also be available to download from the Foundation's website within 24 hours. However, webcast slides are also available right now if you want to download them and then follow it during the presentations. To do that, under the event resources, there's a PDF copy of the presentation. You can just go ahead and download it if you want to download it right now. Recently, biofiltration has been a topic of interest for a number of Research Foundation subscribers. Recognizing this, the Foundation has funded the Biofiltration Research Area in 2012 with a goal to determine biofiltration effectiveness at removing multiple contaminants, define benefits and communicate to key stakeholders, and provide utility guidance on optimizing biofiltration. Foundation to date, under this research area, funded 6 projects. 5 of those actually completed. The last project, which is the guidance, biofiltration guidance manual, is going and is ongoing, and it's going to be completed sometimes later this year. Today, we are going to hear about guidance manual for monitoring biological filtration of drinking water. Under this research area, Foundation has spent over $1.5 million. However, total of value of this project was close to $4.6 million. Right now, I would like to introduce the presenters for today's webcast. We have 2 excellent speakers lined up for today's webcast. Jennifer Hooper is professional engineer with 13 years of professional experience focusing on research and development of innovative biological and chemical treatment technologies. She has an MS from Cornell University in environmental engineering. She has been involved in over a dozen projects for the Research Foundation, Water Reuse Research Foundation, and DoD. She has been a lead engineer, co-PI, PI, on 7 Research Foundation biofiltration projects. She leads water and wastewater related projects at CDM, Smith's Environmental Research and Testing Laboratory in Bellevue, Washington. Our second speaker is Christina Alito. Christina is a water and wastewater process engineer in HDR's Northern Virginia office. She obtained her PhD from Duke University in environmental engineering in 2012. With a focus on the effects of heavy metal contaminants on biological nutrient removal processes. Since then, Christina has been heavily involved in biofiltration process optimization. And then she was also involved in the Water Research Foundation projects 4555 and 4620, which is the webcast for today. Christina is the chair of HDR's Drinking Water Compliance and Technology Practice Group, which connects HDR professionals and fosters discussion of emerging water quality issues. With that introduction, I'm going to turn the microphone to Jennifer. She's going to start out with the presentation. Jennifer, it's all yours.
Okay, thank you. Well, welcome everyone. I'd like to also give a shout out to Chance Lauderdale from HDR, who was a co-principal investigator on this project, as well as Lauren Weinrich from American Water and Orange Schneider, who provided some significant contributions to the project. So just want to recognize those folks as well. So to start off the presentation, I'd like to cover the definition of biological filtration that was developed by the AWWA Biological Drinking Water Treatment Committee. And they define biofiltration as filtration with the operational practice of managing, maintaining, and promoting biological activity on granular media in the filter for enhancing removal of organic and inorganic constituents before treated water goes to the distribution system. So biofiltration is not a new practice, not a new technology, but rather an operational practice of filtration where we're intending to enhance the biological removal processes occurring within the filter. And here we show some pictures of an anthracite filter. We show the full filter bed here on the left. The middle picture has a picture of the filter media for a biological filter. And if you look at it, you see it looks like filter media from a non-biological filter, a conventional filter. And that's because the biomass that's on the surface of the filter is typically very small. The biofilm depth is usually 10 micrometers to up to a millimeter or so thick. So it's typically not visible to the naked eye. Instead, if you look under a microscope, you can see bacteria populated on the surface of the media. And here we're showing an electron micrograph, scanning electron micrograph of the bacteria in a biofilm here on the right, this photo on the right. We could see some rod-shaped and some cockeye bacteria, as well as the extracellular polymeric substances, or EPS, which help provide the glue by which bacteria can attach to a filter grain surface and also comprise the matrix, the biofilm, of the biofilm. So how do we monitor for biological filters? Well, as Kanan mentioned, the Water Research Foundation has provided a substantial amount of funding to help propel our understanding and knowledge and research in this area over the past 10 years. There was an earlier project that CDM Smith and American Water collaborated on called a Monitoring and Control Toolbox for Biological Filtration. And from that project, we evaluated 40 different monitoring and control tools and provided guidance on those tools that are most highly recommended for use for biofiltration monitoring. But in the process of going through that project, there were some new innovative tools that we felt needed some additional characterization and further vetting to really provide a more robust recommendation on how they should be implemented. So that was really the impetus for this current project that we'll be discussing today, which was originally called a bioprocess monitoring tools for biofiltration. And the focus of this project was to provide guidance on what to monitor for, how samples should be collected, what values are acceptable, and really providing the context for data interpretation. And the results from this project are being integrated with other Water Research Foundation projects like the Biofiltration Guidance Manual for Rapid Rate Filtration Facilities. This is a project that's currently underway, and the final deliverable for that project will also feed into the manual of practice that the AWWA Biological Drinking Water Treatment Committee is developing for biological filtration. So there's multiple different efforts going on, but we're all collaborating together to provide a robust evaluation of and recommendations on how biofiltration should be monitored and managed. So for our current study that we'll discuss today, we organized our project into two phases. The initial phase was to conduct some method refinement sampling. And here we were trying to understand some basic questions on how samples should be collected, handled, and analyzed in the laboratory. This was a one-time sampling event at 5 utilities. And we were trying to hone in on some specific questions, like, for example, should you collect a sample from filter media? If you're collecting that sample, should it be before or after a backwash? If you're collecting that sample, how long should you hold it before it needs to be analyzed? For example, ATP or extracellular polymeric substances. There was some data gaps in the literature on how long those samples should be held before they need to be analyzed. So those are just some specific examples, but we'll go into details as we go through this presentation. Another key aspect of that first phase of testing was to evaluate method reproducibility, and interlaboratory testing. We wanted to understand better what the general variability in the results were, as well as if samples were analyzed by multiple different operators or different labs, what was the comparability in those data. And based on the findings from this initial method refinement and sampling phase, we developed standard operating procedures that were used as the basis for our test plan for tool validation testing. And this was conducted over a 9 to 10 month duration at 15 facilities. And the location of those facilities is shown on this map here on the right. Those facilities included both full-scale plants and 2 pilot plants. The nice thing about incorporating 2 piloting facilities was we were able to conduct some specific controlled experiments that might not lend itself quite as well to a full-scale facility. Here we looked at one test was to enhance microbial activity by intentionally adding additional substrate, a biodegradable substrate, over a specific duration of time. And the second experiment that we conducted was to intentionally attenuate bioactivity by adding a disinfectant to the filter influid. And the objective of conducting these experiments was to evaluate the response of our monitoring tools to these changing conditions that we were modifying on the filters. Another objective of this study was to further refine the method reproducibility and interlaboratory testing of some additional methods that we weren't able to incorporate with the initial phase of testing. This slide shows an overview of the participating utilities. There were 16 total utilities. 5 participated in the first phase and 15 in the second phase. But what you can see here is we have a broad cross-section of water sources, flow rates in terms of the design size of the facility, different media types. We had sand, anthracite, and GAC filters, as well as different types of oxidants that were used upstream of biofiltration. We even had a utility using UV hydrogen peroxide, and we had 2 utilities that didn't have any upstream pre-oxidation at Greater Cincinnati Waterworks in West Palm Beach. They didn't have any pre-oxidation. We also had facilities with different backwashing strategies. It's not shown on this graph, but it is in our report. We had chlorinated, unchlorinated, and chloraminated backwashing strategies. So the intention here was to try to capture a wide range of conditions because biofiltration is used in multiple different contexts. We wanted to make sure that the data we were acquiring from the tools captured that wide degree of variability. This slide shows the monitoring tools that were included with our study. And today's talk is going to primarily focus on the biological monitoring tools. And those include the biofilm formation rate, dissolved oxygen consumption, adenosine triphosphate, or ATP, adenosine monophosphate, or AMP, extracellular polymeric substances, or APS, and microbial community analyses. The other tools that are shown here for characterizing organic carbon, the natural organic matter, as well as water quality and filter hydraulics and operational parameters. These components are also very important for understanding the process. But the primary focus of today's talk is going to be on the biological tools. One key item you might notice that's missing from our list of organic parameters is AOC and BDOC. These constituents are commonly included in research studies. Assimilable organic carbon is AOC and biodegradable organic carbon is BDOC. However, these 2 fractions of organic carbon are not particularly practical for regular monitoring and operations. That really was the objective of our project, was to evaluate tools that had high data quality, were cost effective, reproducible, as well as operator friendly. And so AOC and BDOC, they just have limitations on their cost and their turnaround time, and some issues with data quality in terms of sensitivity and variability of results. So we did not include either of those in this current study. So the first tool I'd like to go into detail on is the biofilm formation rate. So this is looking at the growth of biomass on the surface of coupons over a designated period of time. So the way it works is you install coupons into a coupon holder, and these coupons are the same kind of coupons that you would use for corrosion monitoring and distribution system, except that we used polycarbonate as the material type instead of a metal. An example apparatus or installation of these pipe loops is shown in a picture here on the right. This was installed at Columbus Water, and it's showing flow going through the system on the lower right-hand side of the picture, moving right to left, where flow goes in past a manual control valve, then it goes up past a sight tube, and then past the coupon holders through a flow control orifice, and then the effluent goes to drain. Now we had a couple different iterations of this installation. So some facilities used a flow control orifice to control the flow rate at approximately 0.5 GPM. Other utilities used a peristaltic pump or a chemical metering pump to help control that flow rate, and other facilities use a manual control valve. The key thing is to keep a consistent flow rate across the surface of those coupons during the incubation period so that you don't get stagnant flow. We also don't want flow to be so low that you start getting issues with particle deposition through the pipe loop. So it needs to be high enough and consistent enough to maintain flow through this pipe loop apparatus. And these pipe loops are 3/4 inch diameter, Schedule 40 PVC pipe. They're relatively cheap to install. It's about $60 to $100 or so in parts. And then the coupons are $16 a piece. And after you incubate the coupon for a particular period of time, you extract it and measure ATP as an indication of the biomass that has accumulated on the surface of that coupon. When we install this and measure the biofilm formation rate at the biofilter influent, that's an indication of the rate of biological growth or potential bioactivity that's going onto the filter influent. This is the water quality that has the highest substrate going onto the top of the filter. So it's your highest biological activity, you could say. If we measure it at the filter effluent, that's more of an indication of biological stability in terms of how much biological growth you get after the biofiltration process. If we look at the change in the biofilm formation rate from the influent to the effluent, that's an indication of biological activity as well. So this slide has a little more detail on the procedure. Again, we're installing polycarbonate coupons. We incubate the coupons for a duration of 2 weeks and then remove the coupon and place it in the Luminultra Ultralight 7 extraction tube. This was for the ATP test kit that we used, which was through Luminultra using the deposit surface analysis kit, the DSA kit. There are other vendors who supply ATP analysis test kits, but this is just the one we used for our project. The nice thing about the Ultralight 7 extraction tube is once you place the coupon in there, it lyses the cells, extracts the ATP, suspends it in solution, and keeps it stable for up to 24 hours at room temperature. If you need to wait a longer time period before analysis, you can hold it in the fridge at 4 degrees Celsius for up to a week and still get similar results. So after we measure the ATP, then we calculate the biofilm formation rate by dividing the ATP concentration by the coupon surface area and the incubation time. So you might be asking, so how long do we leave it in for? Well, we did an initial evaluation during the first half of our validation testing study to answer that question. Here we sampled over 4 months, so we collected samples monthly over a duration of 4 months from 8 utilities, and we incubated the coupons for 2 weeks and 1 month because we wanted to see how the biofilm formation compared. We wanted to make sure we had enough growth on the coupon surface that we could get a measurable reading, but we also didn't want to leave it in for so long that we move into a decay phase on the surface of that coupon, meaning the biofilm is fully saturated over that surface and you start getting sloughing of biomass. So we wanted to make sure to capture that growth phase. And based on this data, you can see if you look at a couple of data points, the concentrations were of the picograms of ATP per millimeter squared were pretty similar regardless of incubation period. In a couple of cases, the concentration increased a bit. In other cases, it decreased a bit. But based on these results, we decided to go with a 2-week incubation period, again, so that we could target a growth phase of the biomass accumulating on the surface of that coupon. When looking at the concentrations of the biofilm formation rate at the filter influent and effluent, we saw we tested this at 8 facilities, and we saw concentrations at the influent were approximately an order of magnitude higher than the effluent in most cases. The biofilter influent concentration ranged from 1.3 to 130 picograms of ATP per millimeter squared. On the effluent, it ranged from 0.93 to 12 picograms of ATP per millimeter squared. Now, an exception to this was at Swimming River. You can see in this graph here, the concentration was actually higher on the effluent than on the influent. It increased by almost double. And the reason the influent was so much lower was because this particular facility, they have chlorine, permanganate, and ozone that's added upstream of their biological filters. And they had 0.6 milligrams per liter of chlorine residual on average during the duration of testing that we ran this for. And they have deep bed GAC filter media. So that chlorine residual that goes onto the filter influent is quenched in the upper portion of the GAC bed and is removed. And the biofilter then has higher biomass as they move through the depth of that filter profile. So this biofilm formation rate measurement here being lower on the influent than the effluent was an artifact of this oxidant residual on the filter effluent and really not a measurement of that facility's biological activity. So a way to mitigate this would be to either have a quenching agent added to the initial portion of the pipe loop to quench that chlorine residual before it goes across the coupons. Alternatively, instead of installing it at the filter influent, the biofilm formation rate pipe loop could be put on a sample tab from a location deeper within the filter bed on the side of the filter profile where there would be no more chlorine residual. Another question we had was, so what do these numbers mean? We are seeing some significant removal, which is a good indication that these filters are biologically active, but what concentration on the filter effluent is acceptable? So for that, we compared our results to an earlier Water Research Foundation study that CDM Smith conducted with American Water that was led by Marc Le Chevalier, where we were evaluating biostability and distribution systems. And here we were looking for providing guidance on methods for assessing biostability. And we included an evaluation of the biofilm formation rate at the point of entry in the distribution system, as well as various locations with shorter and longer hydraulic residence times throughout the distribution system. So we could see how varying chlorine residual or concentrations at the point of entry of the distribution system versus throughout the distribution system impacted the growth of bacteria on these coupons. And what we found was that at the point of entry to the distribution system, so this is after disinfection, a target of 0.09 picograms per millimeter squared per day for chlorinated systems and 0.017 picograms per millimeter squared per day for chloraminated systems was important for the biofilm formation rate to maintain disinfectant residual stability and biostability in the distribution system. So while the filter effluent measurements that we did for our current project was before disinfection, this gives us a good framework for what kind of target concentrations we need to get to after disinfection to maintain biostability in distribution systems. This slide is showing the biofilm formation rate percent removal across filters, and you can see we typically had about 1 log removal of the biofilm formation rate, at 92% or higher removal. There were a couple of exceptions to this rule. In particular, at Halifax Water, the average percent removal was 23%, which is quite low. And this particular facility, they're located in Nova Scotia in Canada, and they have pretty cold water. For the first half of testing, their temperatures were less than 5 degrees Celsius, and their DOC removal was 0.2 milligrams per liter. It's pretty low. And from earlier studies we've done with them, we knew their AOC and BDOC concentrations were relatively low. So we knew in general that at Halifax Water, they were operating as biological filtration, but their bioactivity was relatively low compared to other facilities. So this is another line of evidence that the biofilm formation rate was providing a good indication of biological activity. When we compare the influent biofilm formation rate to temperature, we saw there was a correlation with temperature where higher temperatures correlated with higher influent biofilm formation rate, particularly at a threshold temperature of 15 degrees Celsius. We saw it really take off. And this was good to see because we saw that same threshold temperature dependence on biofilm formation rate when we were evaluating biostability in distribution systems. There was a statistically significant increase in biofilm formation rate at temperatures greater than 15 degrees Celsius. So it was good to see that we were observing similar results. When comparing the biofilm formation rate to DOC removal or substrate utilization, we did see a positive correlation or positive response between influent biofilm formation rate and DOC removal. And this makes sense. If you have more biological activity, you should see greater DOC removal. Now there is a fair amount of scatter here, but in general, we are seeing that positive correlation. There was one outlier, and that was at the green dot shown at Greater Cincinnati Waterworks. They had a little higher influent biofilm formation rate than we would have expected given their DOC removal. But when we look at this facility in particular, we saw that they did still have a positive correlation between influent biofilm formation rate and DOC removal, especially when the temperature was elevated above 15 degrees Celsius. So again, there's a little bit of scatter here, but there is a correlation in terms of higher bioactivity and higher DOC removal. So this is important when we're trying to understand the process and make sure that we're characterizing biological activity accurately. The next tool that we evaluated was dissolved oxygen consumption. And here we used online luminescent dissolved oxygen probes to quantify biological activity. Oxygen is consumed during substrate utilization when organic carbon by heterotrophic bacteria, when that's degraded, oxygen is consumed and CO2, water, and energy are released. So we use this as an indication of biological activity. The probe can be installed in a flow-through cell. It can be pole-mounted and placed at the top of the filter bed in the water above the filter bed or in a stilling well. And from our testing, we recommend installation in a stilling well if possible. For the filter influent, this is just because it allows the probe to be as close as possible to the actual filter influent. For the filter effluent, it can be installed in a flow-through cell. When you install it in a stilling well, though, just make sure to include perforations, little holes along the through the stilling well itself, so that water can mix between the main portion of the water above the filter bed and the interior of the stilling well, so that your DO measurements are, recording based on water quality that's representative of the filter influent. We did find that the limit of detection for DO consumption was 0.1 milligrams per liter. That's shown in this graph here on the lower right, where we graphed the weekly average DO consumption coefficient of variation versus the weekly average DO consumption. And essentially we saw a lot more scatter in the data when there's much lower DO consumptions, and that means you're really approaching that limit of detection. Regarding the upper limit, we had some questions on that since the probe displays, or the controller displays 20 milligrams per liter as an upper limit. Any concentration that's higher than that is just reported as 20 milligrams per liter. But if you download the data from the controller, it actually records concentrations that are a lot higher. So we did some testing, I'll show that in just a second, but confirmed that the upper limit is 43 milligrams per liter. So here's the accuracy testing that we did for the DO probe under supersaturated conditions. This is particularly important because at ozone plants, the DO concentrations can get quite high. And so we wanted to confirm that the DO readings that we were attaining were representative. So the first thing that we did was measure the probe in air-saturated water. This is the methodology that's used for calibrating the probe. And then we subsequently put the probe in 99% pure oxygen-saturated water. So this could get a much higher DO concentration. We wanted to see if the measured DO concentrations were in line with the expected DO concentrations given the temperature and elevation data from the facility we measured this at. And we saw the relative percent difference between measured and expected values was 2.7% for air-saturated water and 0.9% for 99% oxygen-saturated water. So we knew that the upper readings of dissolved oxygen, these high concentrations higher than 20 milligrams per liter, were accurate. So then we installed the probes at 8 facilities, and 6 of the 8 used ozone upstream of their biofiltration process, and we found the concentrations of filter influent DO were quite high, as high as 32 milligrams per liter, which was a little surprising. When we looked at the change in dissolved oxygen from the filter influent to the filter effluent at these facilities, we saw very high DO consumption values that were much higher than what could be calculated based on the theoretical oxygen demand from carbon utilization. So it was far in excess of predicted values. It was like 18 milligrams per liter of DO removed. So so we concluded from that there are other mechanisms occurring in terms of physical equilibration processes from the supersaturated dissolved oxygen in the filter media, either from air binding or just off-gassing of oxygen, that was not representative of biological removal processes, and therefore DO consumption should not be used for pre-ozone facilities. However, when we installed the probe at non-ozone facilities, we saw a very good correlation between the theoretical oxygen demand and the measured DO consumption. So here I'm showing the equation for calculating the theoretical oxygen demand based on the DOC removal, and we've plotted the DO consumption in blue and the calculated theoretical DOC removal in as the black dashed line. If we compare that black dashed line to the actual DOC removal from grab samples shown in the black dots, you see it's pretty close. It's not exact, but it's very close in terms of predicting the values. So that's another line of evidence that we are indeed measuring biological activity, and it is related to substrate utilization. The next thing that we compared was our biofilm formation rate readings with the DO consumption. And instead of just comparing it as the removal of or change in DO from the filter influent to the filter effluent, we calculated the volumetric DO consumption. And this is because the biofilm formation rate is a rate. It's the rate of accumulation of biomass over time. So we want to compare that to a rate in terms of the kinetics of DO consumption across the filter. So you divide the DO consumption by the empty bed contact time, and that gives you the volumetric DO consumption. And here we saw definitely a positive correlation between influent biofilm formation rate and volumetric DO consumption at these 2 facilities that did not have pre-ozone. So again, another line of evidence that we're getting consistent information in terms of biological activity. A controlled experiment that we did at one of our pilots was to look at addition of a substrate. We added 2 milligrams of carbon per liter of ethanol, and we looked at how over a duration of 2 weeks, and during that time we looked at how much additional organic carbon was removed and how much DO was consumed. And we saw when the 2 milligrams per liter of carbon was added, we saw an increase in the organic carbon utilization up to, organic carbon removal up to 0.92 milligrams per liter. And commensurate with that, we saw an increase in the DO consumption across the filters over that time period. It took about 72 hours for the DO consumption to reach approximately steady state conditions. But as soon as we turn that the ethanol pulse off, the DO concentrations rebounded to baseline conditions instantaneously. So that was a clear indication that we were getting a measurement here that was reflective of known changing conditions on that biofilter. When we looked at inhibitory conditions, we added a chlorine spike, and here we did a pulse of 0.5 milligrams per liter for 24 hours, and then we did another pulse of 5 milligrams per liter for the next 24 hours. And here we just wanted to see if we did a small change in inhibitory conditions and then did a very big change, what would a dose-response relationship look like? And these were on anthracite filters, so we didn't have any quenching of oxidant residual due to GAC media. And we saw the DO consumption definitely declined as the chlorine spike increased. However, the DO concentrations were quite low to start. So I mentioned earlier about the Halifax water pilot that they had relatively low bioactivity. So it's not a perfect data set, but we did see a decrease in the DO consumption when we added the chlorine spike. The next tool that I'll discuss is adenosine triphosphate, which is ATP, a bioenergy molecule in all living organisms. This is really an indicator of biomass rather than bioactivity, and I'll show you some data why we think that in just a minute. But there is some confusion in how it's conveyed at times in terms of whether or not it's bioactivity or biomass, and so that was one of the things we tried to help elucidate as part of this project. One of the first questions we had was, how do we collect the sample? So here we collected ATP on the filter media as opposed to in aqueous samples. The ATP concentration on filter media tells you how much biomass is on your filter media, and that's the biomass that's actually doing the work in terms of degrading contaminants, as opposed to the aqueous phase ATP, which is not responsible for contaminant destruction. So the first thing we wanted to evaluate was how long can the sample be held before you start to see changes in ATP. And what we learned was after approximately 48 hours, you start to see decreases that are substantial. After 1 week of holding at 4 degrees Celsius in the fridge, we saw a 10% decrease in the ATP concentrations. So from this work, we recommended a 48-hour hold time. We also tested whether or not samples should be collected before or after a backwash, and we recommended before a backwash, and this is because, especially for filters that have chlorine or chloramine in their backwash, there is impacts from that. We see a little lower ATP concentrations on the filter media after a backwash. In particular, there was one facility, Denver Water, where they had a very high chlorine residual in their backwash. It was 10 milligrams per liter. That's very high. And we saw some significant decreases in ATP right after backwashing due to that exposure to disinfectant. The other question we had was whether or not composite samples should be collected. If you collect a sample from one portion of the filter bed, is it going to be the same as a different portion of the filter bed? And we found that they were relatively consistent and that a single grab sample was sufficient. When we look at ATP on filter media, we see there was some correlation with temperature in terms of facilities that had higher temperatures throughout the year did have higher biomass, but if you look at the same facility, their biomass was relatively consistent regardless of temperature. So it was not a direct correlation with temperature at the same facility. Similarly, with organic carbon removal, there was really a lot of scatter in the data and no direct correlation there. So this is another indication that ATP is measuring biomass as opposed to biological activity, because we know bioactivity is influenced by both temperature and substrate utilization. The next test that we did was testing ATP on filtered media during our substrate loading test and our chlorine spiking test. And these were, again, intended to enhance activity or inhibit activity. And we saw when the substrate loading test was conducted, we saw no change in ATP, both at the Gwinnett County pilot and the Halifax water pilot. However, when we added chlorine to the filter influent, we saw some significant decreases in ATP, both at the Gwinnett pilot and the Halifax pilot as well. And so what we learned from this was that ATP, again, was an indicator of biomass, where if you add disinfectant, you're going to slough off and kill your biomass. But if you add substrate, which would enhance biological activity, there really was no change in ATP. The last tool that I'm going to talk about is adenosine monophosphate. And this is a new test kit that has been developed by Lumen Ultra for the oil and gas industry, and they were using it to characterize bioactivity for assessing the degree of biofouling in fracking wells. And why this is helpful is it gives you an indication of the relative energy state of bacteria. So this is an indication of biological activity. AMP is adenosine monophosphate. That's the lowest energy state of the adenosine phosphate molecule. And adenosine triphosphate has 3 phosphate functional groups. That's the highest energy state. So when you take the ratio of AMP to ATP, that gives you an indication of the relative biological activity. So we tested this at 2 facilities. One facility we tested for 4 months, and the other we tested for the full duration of the study, which was 9 months. And I'm showing that data here. That was at the Halifax water pilot. And we saw some interesting trends for when the temperature was greater than 15 degrees C, which was actually only a few months in the summer. The DOC removal increased. When we looked at the biofilm formation rate, the influent biofilm formation rate increased, and the biofilm formation rate net removal increased at higher temperatures. And when we look at the AMP to ATP ratio, it decreased at higher temperatures, which is exactly what you would expect. If there's lower AMP, that means there's more bioactivity. So we concluded from the study that this is a potential monitoring tool that has viability for biofiltration monitoring, but it needs additional validation since we really only had a good data set for one utility. So we do recommend this for further validation in research and optimization studies. And with that, I'll hand the presentation off to Christina to talk about EPS.
Thanks, Jen. So I'm going to talk about two more tools that we looked at. The first one being extracellular polymeric substance measurements. So EPS are excreted by bacteria, and they're used as the matrix to support biofilm growth. Typically, they're made up of 10% microorganisms and 90% of this biomass. And there are multiple functions, but it helps protect the organisms from stress, temperature, and it helps them to collect nutrients as well. So usually we see excess EPS with nutrient limitations. So EPS is comprised of polysaccharides or carbohydrates, protein, and DNA. Now, one thing to note is microorganisms can both produce and consume EPS as they need it. So they use the carbohydrate fraction as the same way that humans do, as a rapidly available energy source, and then they use the protein fraction as a more sustained energy source. There have been several studies that have looked at the percentage of protein to carbohydrate to other components to look at the composition of EPS, and it's been pretty well documented that the predominant composition is the protein fraction at somewhere at 90%. There are a couple different methods for measuring EPS. We based our method, the modified Lowry assay, using a BCA protein kit to focus on the protein fraction, since that is the more dominant fraction. And it's based off research conducted at the University of Texas by Dr. Keithley and Dr. Kiersitz, where they evaluated 6 extraction methods for EPS. During their study, they also found that proteins were the dominant fraction from about 11 full-scale biofilters where they sampled EPS concentrations. During our method refinement stage of the project, we did look at the protein to carbohydrate fraction of EPS, and we found the same result. This is data from 5 different utilities that were part of the study. They were all full-scale biofiltration processes, and so what we saw was that the relative fraction of protein was still quite a bit higher than carbohydrates. So we decided to focus solely on protein measurements for characterizing EPS in our study. Really briefly, I'll just describe what the process was like. What was key from this extraction method was that we took about 2 grams of wet filter media from the top of the filter that were collected below this Schmutzsteger layer. So within the top 6 inches of the filter bed, we then ran the extraction buffer protocol that's part of this modified Lowry assay, and then used the BCA protein kit to extract the portion of proteins that were found in 1 gram of the sample. Sorry, my slides are getting moved around. Okay, so the concentration of protein was then normalized to 1 gram of total solids, which is based on the dry weight of filter media. So this slide is just showing an evaluation of the holding time versus the before and after filter run as well. So we looked at the EPS concentrations analyzed within 1 day, within 2 days, and within 7 days. And we did see a degradation in the EPS concentrations over a 7-day period. So we decided to use a 24-day or 24-hour turnaround so that we saw minimal change in the EPS concentrations. Also, we looked at collecting samples at the beginning and the end of a filter run to determine what type of difference that made, if you should stay consistent with what time of the filter run you measure EPS. What we found was that only 1 sample did have a big difference. That was at Greater Cincinnati Waterworks, where we did see that 1 increase. But other than that, we saw pretty good consistency before and after the filter run, so not a lot of variability in the EPS concentrations. It's possible that a substrate loading event could create a nutrient scenario where you would have elevated production of EPS during a filter run that could result in a substantial gain. But in our results, for the most part, we did not see that occur. Now, what EPS has been used for in the past for biofiltration studies has been to look at nutrient deficiencies and also to see if excess EPS production correlates with an increased head loss in the filter. Because as the gunk builds up in the biofilter, theoretically it should be taking up more pore space and contributing to head loss accumulation rate and the filter run times and just impacting the hydraulics. What we found when we looked at EPS concentrations versus head loss accumulation rate and unit filter run volumes was no direct statistically significant relationships between EPS and head loss accumulation rate. And we did look at both individual utilities and this diagram that these figures that have all of the compiled data for each utility. We did see at 1 utility that there were at Delaware River, we did see a positive relationship between head loss accumulation rate and EPS concentrations, but we weren't able to show statistical significance with that positive relationship, and so we do not think that should be recommended, that EPS should be recommended as a tool to be used for tracking with filter hydraulics. Just to get a little bit deeper into that, we did try to see what relationship there was between pore space occupied in a biofilter to EPS concentration to see if the differences in the filter design, if it's a GSE filter, and the porosity and the size of the particles had an impact on the pore space that was available for EPS growth. Now, this part of our study was based off a paper by Moncler, which stated, it's a 2004 paper that states that a greater than 7% occupation of pore space by EPS is likely to be representative of a highly clogged filter. But it should be noted that that is just for a slow sand filter. So we're talking about rapid rate filters, which could have higher clogging rates, and we wanted to see if we could visualize that same reduction in pore space and correlation to EPS. When we looked at the data for the utilities that participated, it was really kind of all over the place. An average pore size occupied was about 1.74%, so it was less than the 7% that was reported in that study, but our standard deviations were also pretty high. So based on these findings from the investigation, we do not think that EPS should be regularly monitored because we did not find that direct correlation with filter hydraulics. So I'll jump into the last tool that I'll be talking about, which is microbial community analysis. We evaluated a few different microbial community analysis tools to be used in the study, and we ultimately landed on next generation sequencing to be able to characterize what the microbial community looked like and its presence in abundance. So we used a tool known as Illumina next generation sequencing. There are several types of sequencing processes that have their own pros and cons that I won't go into today. Some have higher detection. So it's not to say that this tool could not be useful, but this was the one that we ended up landing on. To do this study, we took samples of the media, and then we extracted DNA from those samples, and we amplified a region of the microbial community known as the 16S ribosomal ribonucleic acid gene. This gene represents all bacteria that have been identified. So when you amplify this gene, you're amplifying the bacterial community in your sample. From that, you can sequence the data, and you can get taxonomic data from the different types of microbial organisms that are present. And I think it's important to point out that the level of detail we were able to get was phylum and genus level data. So as a reference, if you've ever seen E. coli, the word E. coli or Pseudomonas aeruginosa, the first word is the genus level. The second level, the aeruginosa, is the species level. So we were not able to obtain species level information. That can be really critical when you want to look at pathogen identification, and even going beyond that, there are specific types of species that are related to pathogens. So we were not looking at pathogens. We were only looking at diversity of our samples. Now when you do a study like this, you get a ton of data, and we compare data from 2 labs. And what you see is basically the abundance, so the percentage of a particular genus or phyla that's present in your sample. So I'll show you what that data looks like. This is a lot to look at, but what you're seeing here are stacked bar charts, and each different color represents a different organism that's present in the sample. You'll see 2 labels, MD and UT. MD was a microdetectives lab. It is a commercially available lab where you can send a sample. They send you a box, and you put a sample in it, and you send it back to them, and they do 16S characterization with a proprietary method that also uses the same type of sequencing to tell you what is in your sample. We also sent samples to the University of Texas and tried to match our protocols as best that we could to compare the results between the labs. So what I want to show you from this particular diagram is that for the 3 utilities that are shown here, Fairfax Water, Columbus, and Gwinnett, we saw pretty high variability between seasons, and in most cases in the fall when there were lake turnover events, we did see pretty big jumps in diversity. And there were a few key organisms here, such as Bradyworhizobium, which has been, as part of nitrogen fixation, has been very well identified in biofiltration in previous studies. When we focus here on the on the field duplicates, you'll see there's a sample called Spring and then Spring duplicate for microbe detectives. You do see that there is variability between the field duplicates. Now, building upon that, when we looked at the lab duplicates, so the same sample sent to 2 different labs that had slightly different targeted areas of the 16S gene, we saw really drastic differences in the quantification and abundance of organisms. And so, if there's anything you can take away from this, it's if you're going to do this type of analysis, I highly recommend that it is only done at one lab, and if you could even get the same person to do your samples each time, that would be ideal. But it does help to visualize the differences in the microbial community over time. With all that said, this is another tool that we do not recommend at this time for biofiltration characterization. There are several difficulties with running these types of analysis. First of all, they're pretty expensive. They can range from $300 to $600 per sample. The bioinformatics software is not very user-friendly or intuitive, so it takes some time to analyze the results. And ultimately, I think we're looking more, it is not a tool that's great for pathogen tracking. You can't use it for that. And there are other bioassays such as enzymatic studies or microbiome characterizations that could be more useful for measuring big changes in the biofilter over time. So with that, I'll pass it back to Jen just to wrap up with our biofiltration monitoring guidance.
Okay, thanks, Christina. So again, the focus of this project was to provide guidance on practical tools for operators for use on biofiltration monitoring. So some of these tools that we discussed may have application in research studies or potentially in optimization studies or troubleshooting, but for practical use, especially for regular, normal process conditions, there's some key tools that we recommend for monitoring. The first is the biofilm formation rate and for biological monitoring, and DO consumption. However, DO consumption should only be used at non-ozone facilities. Other tools like ATP, EPS, AMP, and microbial community analyses can be used, but they should only be used in specific instances. EPS in particular for troubleshooting could be helpful for having hydraulic issues. And microbial community analysis might be really interesting to look at, but it should be focused to more of a research study as opposed to, collecting samples on a monthly basis or something like that for regular monitoring. For organic carbon and water quality monitoring, we highly recommend including DOC. A lot of facilities look at TOC. If they do characterize organic carbon, many facilities use TOC, and we recommend looking at DOC because the dissolved fraction of organic carbon is what is used by bacteria for biological processes, as opposed to removal of particulates that can happen in the filtration process, which you would capture with TOC. So would definitely recommend including either both of those or just DOC. And pH is very important for making sure you have conditions that are appropriate for biological growth. So a pH of 6 to 9 standard units is a pretty good framework. Temperature is also really important. We discussed the impact of increasing temperature on biological activity earlier in this presentation, and certainly turbidity in terms of meeting regulatory requirements. When we look at filter hydraulics and operational parameters, our top tools that we recommended, we're looking at head loss accumulation rate for constant flow filters, or surface loading rate for constant head filters, empty-bed contact time. We talked about that a little bit earlier, but it's more discussed in more depth in our guidance manual. But that's really important for biological processes, and a contact time of 5 to 15 minutes is pretty standard for the industry. And oxidant residual. If there is an oxidant residual going on at the filter influent, we typically try to minimize that, especially if you have an anthracite or sand filter, since there won't be any quenching on the filter media. And again, the top recommended tools that we included in our guidance manual are biofilm formation rate, DO consumption, and DOC removal as their indicators of biological activity. pH, temperature, oxidant residual, and empty bed contact time are certainly important for impacting bioactivity, and for looking at filter hydraulics, the head loss accumulation rate or surface loading rate are good tools for evaluating that. And just a quick shout out to all the folks who helped support this project. It's a very big team. I mentioned Chance Lauderdale earlier as the co-principal investigator. And we certainly appreciate all the support we got from our project advisory committee, the technical advisory group, numerous vendors, and participating utilities who helped support us in this project. So thanks to everyone who helped support this. And with that, we can move to questions.
Great, thank you, Jan and Christina. Unfortunately, we are on top of the hour. We were hoping that we would have a few minutes at the end of the webcast to address some of the questions, excellent questions that we received. But given that we are out of our time, what we will do is actually we'll go back to those people that ask questions, and we'll provide the written responses to those questions. So again, my apologies. So our webcast was a bit longer than we expected, but so since we don't have time to ask the questions, again, we will provide written responses. So with that, I would like to thank you, Christina and Jennifer, and then our audience for attending this call. And for our audience, slides and the webcast will be available on our website within 24 hours. And as you are exiting the webcast, there will be a short survey. If you can actually fill out that survey, that would be helpful for us. With that, I would like to conclude our webcast, and thank you for your attention.
Wow.